This webinar is the fourth of 2024 series, is part of our communication campaign, Achieve Better Milk Together. At Vetoquinol, we aim at working hand in hand with our stakeholders to achieve more for vets and farmers, cows and consumers, which means healthier cows and herds, better quality of life, and supporting sustainability of milk production. Beyond products, we are aiming at providing solutions to help you solve your and your farmers' pains in mastitis treatment with diagnostics and fit-for-purpose protocols, in mastitis prevention with products and management tools, and partnership in data management to help you provide attractive services. Our topic today, new developments in bovine mastitis, epidemiology of intramammary infections. Our speaker, Professor Inte Schuken, Chief Executive Officer at Royal Gede in the Netherlands and Professor of Management of Farm Animals at Wageningen University, but also Professor of Farm Animal Disease Control Programs at the Veterinary College at Utrecht University. Dr. Schuken's research approach is based on understanding epidemiology and pathobiology of the disease and population dynamics on infection diseases in animal populations. Inte worked for many years at Quality Milk Production Services, QMPS, at Cornell University in the US. As an expert in bovine mastitis and infectious diseases, he has published extensively in peer-reviewed and public literature. Inte has also consulted on many dairy farms across the world and taught courses to groups of veterinarians, farmers, and students. Inte, the floor is yours.
Thank you, Stefan. Thank you also for the introduction. Yes, I really enjoy this opportunity to discuss with you today about the new developments in bovine mastitis, and I will share my slides with you. I think you can see my slides.
Yes, perfect.
And my pointer.
Yes.
Yeah, so I'm, like I said, I'm very eager to share with you the new developments in bovine mastitis, particularly in the epidemiology of intramammary infections. I'll share some of my own research, but also from others. And I think at this point in time, we have very exciting new opportunities as veterinarians, as practitioners, and also in the world of science. And I hope you will recognize that at the end of this presentation. So when I talk about trying to improve mammary health, when I go to farms to consult, I look at three areas essentially. The first is what is the risk of an infection? Cows are always exposed to bacteria, but what is the risk that they actually get infected? And can we do something about it? So one is hygiene and housing to reduce exposure. The other one is the susceptibility. The cow sees the bacteria but doesn't get a long-term infection. So that's the first one. The second one is to try to reduce the duration of intramammary infection, so to make the duration as short as possible. And two important tools for that is treatment of the existing infections, of the infections we know of, and culling of cows or culling of the quarter, so that she's not participating anymore in the infection dynamics. And the third component is to reduce the risk of transmission. And so a cow is infected and is shedding bacteria, usually in the liners, and thereby other cows can become infected. So preventing transmission to other cows also helps in keeping the herd clean. And for that, the milking procedures and biosecurity are very important components. So the 7 areas, so to say, to focus on if you look at animal or other health on dairy farms are these 7 components. And I will more or less look at some of the highlights at each of these areas. So the first thing I will talk about is essentially the transmission component. That's this one here. How can we reduce the amount of transmission between cows, and particularly the milking procedures and the milking system are important. But also, the first thing is how do we know there is transmission? And for that, I think we have currently very nice modern diagnostic tools available, and I'll discuss those a little bit with you. First, a little bit the wording around it. And what we call environmental bacteria, what we typically call environmental bacteria, are the coliform bacteria and the non-Agalactia streptococci, Streptococcus and Streptococcus discalactia. And what we learn, what the National Mastitis Council fact sheet says, is that the primary source of these environmental pathogens is the surroundings. So from the surroundings, the bacteria go into the mammary gland and cause an infection. Contagious bacteria, on the other hand, such as Staph aureus, Streptococcus agalactia, and Mycoplasma bovis or Mycoplasma species, are the ones that are going from cow to cow. Contagious organisms, according to NMC again, are adapted to survival and grow in the mammary gland and cause infections that have a long duration. And the infectious gland, the gland that has the bacteria, is the main source of these organisms, and the bacteria transmit from cow to cow or from quarter to quarter. And so those are the terms environmental and contagious. And typically we connect bacteria to that. Environmental, E. coli, and the Strep uberis, Strep dis, and the contagious, Staph aureus, Strep agalactia, and Mycoplasma. However, I will show you that that's not the complete story. Because if you look at the concept behind contagious and environment, what it means is that one cow is infected, it goes from udder to udder. One cow is infected, it infects the next cow, and it infects the next cow. And what you would expect is if you look at these bacteria in more detail, that the bacteria that goes from here to here is the same one that goes from here to here and the same one that this cow is infected with. So what you would expect is if you take bacteria from this gland and from this gland and from this gland, that we have an identical genetic pattern. And I'll show you genetic patterns later. So it's all the same strain. In contrast, when we have the environmental infection, there is so many bacteria in the environment that you would expect that every cow, every mastitis cow, will have a different strain. And so this cow has strain B, this cow has strain C, and this cow has strain D. And those strains are not moving between cows. They only come from the environment into the cow. So again, strain typing or genetic identification of the strains will tell us whether we have to do with a different strain in each infected individual. Well, and for that, like I said, we have currently very nice molecular tools available. And that looks a little bit like this. And so this is a DNA pattern where the DNA of a bacteria bacteria is grown up, the DNA is extracted, and then it's cut into little pieces at specific parts of the genome. And if the bacteria are different, if they have a different strain for each infection, then you will see that these genetic patterns are not the same in the different strains. So here we have these 3 strains, strain 1, strain 2, strain 3. And each of these strains is different. This particular DNA blob is not here, this blob is not here, this blob is not here and here. These are identical, these are different, this is different. And if you look at the whole pattern, you say, okay, these are different strains. So even though it's the same bacteria, these are all Streptococcus uberis, every single cow is infected with a different strain, and it meets the classical concept of environmental infections. However, also Strep uberis, but now what we see is that all these 1, 2, 3, 4, 5, 6, 7, 8, 9 mastitis cases have the identical strain. Every DNA blob, so to say, is identical between all these 9 strains. So if we see something like this on the farm, we say, hey, Strep uberis is typically environmental. It's one strain per cow, but in some cases, for some reason, and I'll come back to that later, for some reason, it's also able to transmit between cows. So now we have this outbreak of a single strain of Strabuberus. We call that a clonal outbreak. A single strain of Strabuberus is causing all these infections. And the key, of course, now is that the approach to these two herds, to these two different patterns of infection is very different. In the case of environmental infections, you're looking at hygiene and susceptibility of the cow. In the case of a clonal outbreak, a single strain, you're much more looking at the transmission components, how to improve milking procedures, how to milk infected cows last, etc. So having this information is crucial for doing the right thing on the farm. Well, I'll show you a few more examples. This is Staph aureus. Again, as you can see, all these strains are all identical. Well, that's sort of what you expect with Staph aureus, and that it's mostly a contagious organism. It goes from cow to cow. So you would expect that most strains are identical. In this particular farm, we have lots of strain A, and we have 2 cows that are infected with strain B. However, this is also Staph aureus. It's a different farm. I know this farm, and this is a farm where they are very precise with milking procedures. They're very strict in diagnostics. So every Staph aureus cow is separated out and is milked last. Over the time, they do have a number of Staph aureus cases, but virtually every single case is a different strain. And so here we have much more of an environmental pattern. If they want to do something, they want to reduce the exposure to Staph aureus. For example, by having gloves for the milkers. Staph aureus in a lot of cases is coming from human hands or is transmitted by human hands, and gloves might help to do this. In the case of this contagious system, we're much more looking at milking cows last, trying to identify the shedders, because the shedders are spreading a lot of bacteria and you want to either separate those or treat them or cull them. But you have a very different approach in the environmental issue versus the contagious issue. Well, another one that is sort of logical to see is this farm with lots of E. coli cases. And again, every strain of E. coli is a different strain. Well, clearly this is an environmental organism, so that is what you expect. However, on some farms, you do see a different pattern. And in this case, every block is a cow. So it also means that multiple cases in the same cow have the same strain. This is 2 cases. This is 3 cases, 2 cases. So we have long-term E. coli infections, which happens in some cases. And also, it looks like these infections are transmitting from cow to cow. And so the classical thing to do in E. coli, in this case, is improve hygiene, cleaning the udder before milking, cleaning the udder after milking, and to make sure that there is less exposure to manure-like bacteria. But in some cases, you find this pattern where it's much more a single strain or a few dominant strains on the farm that are also showing evidence for transmission between cows. Another area where you can see that is the Streptococcus agalactia. This is work from Denmark, where they identified more and more cases of agalactia. In our data in the Netherlands, we see the same. You sort of see a resurrection, that's what I call it here, of Streptococcus agalactia. And in the Danish situation, they find 2 or 3 main sequence type, strain types, you can say. And from the literature, we know that this particular strain, 103, is a bovine specific strain. So it likes mammary glands and it transmits easily between cows. Whereas these strains are much more common to humans. Humans have infections with what they call GBS, and the agalactia of the humans, so to say. And most likely, these are much more individual infections. And it is quite possible that these infections are actually transmitted by humans to cows. Approximately 25% of people are carriers of GBS, group B streptococci, GBS. And if they're not careful, or even if you're careful, you can still transmit it to cows. And then you see single cases of Agalactia in a herd of cows that are not necessarily transmitting to others. Whereas you have this strain type, 103, the transmission between cows, or as you can see in this part of the graph, between herds is much more likely. What they did here is they calculated the K statistic, and the closer the K is to a straight line, a horizontal line, the more likely that these are random events. So there is one farm that is in the north of the country and one in the south and the east and the west, and they're not really connected. The more that the line goes away from the horizontal line, like in this case, there is strong evidence, and this is the 95% confidence region. So this is outside the 95% confidence region. There is evidence that this is a clustered situation. Well, this is serotype 1, the one that is here. This is serotype 23, the one that is here. And this is serotype 103, and the bovine specific serotype. And as you can see, this is showing clustering of farms with this particular infection. And so clearly it behaves in the farm, but also between farms as a contagious organism, whereas the other two serotypes are much more individual cases. So the concept of transmission is very important. And I think what is really exciting now, and I'll talk a little bit more about it later, is that we have the tools now to distinguish between environmental and contagious behavior, not so much the name of the species, E. coli versus Agalactia, but the behavior of E. coli and the behavior of Agalactia. And for that we have the treatment procedures, but also a correct diagnostic system. And the new developments in diagnosis of mastitis, diagnosis of bacteria in general, are indicated here. First, we have the Mastitis, that is an amino acid based system to identify the bacterial species, and I'll show you some examples of that. The FTIR spectroscopy, Fourier transformed infrared spectroscopy, that's a carbohydrate based system, and it's particularly used for strain typing. So the examples that I just showed you with strains per farm and between farms can be done very easily with this FTIR system, carbohydrate-based system. And then the most complex of these is whole genome sequencing, where we can, and we look at every single base pair in the whole genome of the bacteria. And based on these sequences, we can also say this is the same strain or this is a different strain. Whole genome sequencing used to be a very complicated system, but now with much more simple systems, actually this system here is the sequencer. So if this is the hand of an individual, it is something that is just as large as your cell phone, actually. It's the nanopore system. And it's also, in terms of costs, things become very doable to get a much more complete picture of the whole genome sequence of bacteria. So all these developments lead to the fact that as practitioners, we have much more access to precise information of bacteria. And I'll go through some examples. For example, for one thing that we have learned from the Mastitis is a much better understanding of the coagulase negative Staphylococci, CNS coagulase negative Staphylococci. In the classical systems of bacterial culture, we identified Staph aureus and the Staph non-aureus Staphylococci, NES actually, or CNS, coagulase negative Staphylococci. In the current systems with Mastitis, we find, for example, here we have approximately 20 different Staphylococci that are identified using the Mastitis. And now we know from a lot of work that is done that these three are really the dominant coagulase negative staphylococci, and particularly Staphylococcus chromogenes. And the blue bars here, these are 21 hertz. The blue bars here is the dominant coagulase negative Staphylococcus. They typically are a little bit less pathogenic compared to the Staphylococcus aureus, the coagulase positive Staphylococcus. But what they can have is an enormous amount of antimicrobial resistance genes. So they can still have an impact on the somatic cell count of individual cows, and if you have a lot of these, also on the cell count of the herd. And then having the information on whether we have a particularly large amount of chromogenes or whether we have a different species, in this case it's the xylosis, which tends to be much less pathogenic, is actually of great value. And so the Mastitis helps us a lot in the CNS, and also a similar picture I can also make for the strep species, where we have a much more precise understanding of the different streptococci. The IR biotyper, the second piece of equipment that I showed you, is used for strain typing. And using a genetic tree, so to say, we can identify whether strains are virtually identical, and they are genetically very much the same, or whether they are different. And in this case, this is a dairy farm with lots of Klebsiella cases. What we see is that we have three types of Klebsiella. Type A, and this is sort of the cutoff of a new strain. So this is a different strain, this one here. This is a different strain. There's a lot of these, strain B, and this is a different strain, strain C. And as you can see, in this case, strain A causes a number of infections, and there is one milk or mammary gland infected, and the other one is from the environment. So this seems to be an environmental strain. But this Klebsiella strain has lots of infections and infects actually 4 different cows, and can also be found in the environment and also in the manure of cows. And we know from Klebsiella that it's particularly present in the manure of dairy cows, and it looks like through that route it is infecting cows. And there is also evidence in this case, 1, 2, 3, 4 glands infected, that there is also transmission between glands. And this one, again, the number C strain, much more looks like an environmental strain. Well, these tests are actually in the area of cost of a normal culture. You first have to culture them, and there is a cost to that. And then to do this biotyper diagnostics is approximately the same cost as doing a culture. So these tools become very much applicable, or very much in the hands of day-to-day work on dairy farms. And by using these tools, you get a much better understanding of what is happening on the farm. Well, the last one, the small piece of equipment that I showed you, the sequencer, provides us with much more detailed information. And this, at this point in time, is not really something that is used in the practice, but still we can learn a lot from this. This is a study that is done by one of my PhD students. He has about 150 different strains. They are indicated here. And she sequenced all these strains, and these are the virulence factors, so the pathogenicity characteristics. If they have a particular gene present, so it's present, this gene is present in these strains, and it's not present here, and then it's present here. And this gene is not present in all these cases, and it's present in a few cases here. So all these pathogenicity genes are aligned with the antimicrobial resistance genes. And then you can see that some strains, these are different strains, again, using the same genetic tree system. And this is one strain, the number 2, this is the number 3. So you can see that certain strains have virulence factors, but have virtually no antimicrobial resistance, whereas other strains have, and this is strain 6, has lots of these pathogenicity genes, but also has a number of antimicrobial resistance genes. And this one, number 4, even more, lots of pathogenicity and also lots of presence of antimicrobial resistance. And so some strains are much more likely to attract, adapt, take on, so to say, the antimicrobial resistance genes compared to other strains. And of course, having the knowledge of this provides us also with more understanding of the biology of infection of these strains. And the final diagnostic tool that we also start to see is the antibodies, either in bulk milk, as shown in these papers, or in individual cow milk. And Mycoplasma bovis is of particular interest. It's a not so easy bacteria to culture, and the antibodies tell us something. This is data from Sweden, this is data from Ireland, and in both cases, what we see, and the dark red ones here, or the dark points here, means there is bacteria presence, and the darker the red color is, the larger the herds. So as you can see, there is a tendency for large herds to have much more mycoplasma to compare to small herds. And it's most likely because of the transmission dynamics. To be successful as a bacteria, you need to have a certain amount of animals present in the farm, and larger farms are more susceptible to transmission issues compared to small farms. And also, we see that in the iris data, the herd size is a very significant risk factor for infection. And also the number of close by neighbors that are infected is also a significant risk factor. And so again, herd size, transmission characteristics seem to be important for the likelihood of infection with Mycoplasma bovis. Well, my point is, I go to farms, and if you look into a little bit more detail using these new tools, there is so much more to learn and so much more precise methods to use that it's also attractive. It's also helps you in coming up with the best prevention program on the farm. Well, as I mentioned before, one of the 3 components to look at is the duration of infection. And by using treatment, you can, of course, reduce the duration of infection, because treatment essentially has 2 effects, what I summarize here. First is the direct treatment effect. You treat a cow and that cow will cure. So it helps the cow directly. But there is also the indirect effect. And that means that if you treat a cow, she's not shedding anymore. So the rest of the cows in the farm are actually more protected. And so treatment has 2 components. One is the direct effect, and the second one is to have an indirect effect. And we studied that in using some molecular or some mathematical methods by using what we call a state transition model. So we looked at modeling infection-free cows. Then these cows become infected. They have a short-term infection. Then the infection remains, and they have a long-term infection. And if animals get treated, if you treat either the short-term or the long-term animals, you get cured animals. Of course, cured animals can also get reinfected, and then the whole cycle starts again. So by using this relatively simple model, we were able to show that treatment actually has these 2 components. But it depends on the transmission ability. And the transmission ability means how likely is that an existing infection is causing a new infection, either a new infection here or a reinfection here. And it turns out that actually this transmission component is very important. And that is what's shown in this graph. If there is very little transmission, then what treatment does is the direct effect. It treats the infected cow and it leads to cure. If there is some transmission in the farm, then the treatment of cows also helps in reducing new infections. And so the direct and the indirect effect leads to the overall efficacy on the farm to almost double. So if you treat cows on a farm where there is relatively limited transmission, then you have a very effective treatment effect. However, if the transmission goes very high, and so there is lots of chance that these cows are infected, then treatment becomes less and less useful. And because a cow may be treated, but she gets immediately reinfected. And therefore, on the farm itself, the total treatment effect is virtually disappearing. So the lesson to learn here is that as we know that transmission is so crucial, it's one of the three factors that I started with, but it also has an enormous impact on the herd efficacy of treatment. So therefore, first reduce transmission, move this way with the transmission parameter, and then what you will see is that treatment is actually quite an effective way of limiting the total intramammary infection problem. And there is, fortunately, a lot of good studies on treatment, comparing treatment to controls or comparing treatment procedures. An interesting paper recently that I saw was the descriptive study on how animals are treated in the different countries. I won't go through the whole table, but it's quite surprising to see that in different countries there is very different treatment regimens. Even though we have an international scientific community and international scientific papers, we still see very specific uses of treatment per country. So I think that's, and knowing of course from which country you are, you'll see that there is a specific treatment characteristics per country. Well, the third component, the final component that I would like to discuss is the susceptibility to mastitis. And so we talked about reducing duration, we talked about transmission, and this is the risk of intramammary infection. So how can we reduce the risk of infection? Well, first of all, there is the immune components. One is the existing component, the innate component, the anatomy and the cellular systems and the bacterial ecosystems. And then there is the adaptive component, so the antibody dependent or the memory dependent component of the immune system. Well, let's go through examples of both components. The innate component is the inborn component, so to say, and one of that is the T-dens of the cow. And we know that very rough T-dens, so T-dens like this, have a much higher likelihood of infection. And this is a recent study, or a study from the group from Bristol, showing that cows with very bad T-dens have a higher likelihood, approximately 2 times higher likelihood of infection. Well, with milking machines, with liners, etc., you can deal with very bad T-dens. Another component is the likelihood of infection during the dry period, and particularly the milk production at dry off. And so how much milk the cow is giving at the time that you milk her last is a good predictor for intramammary infection during dry off, during the dry period. And so the higher the milk production, the higher the likelihood of infection. Most likely the T-den is not very well closed and bacteria have an easy time to go in. Most likely there is more milk leakage in the high production animals. So reducing milk production is a very effective way of improving the dry cow period. Another one is the lactoferrin. Lactoferrin is binding the free iron, and the free iron is used by bacteria for multiplication. So by improving lactoferrin or faster increase of lactoferrin, you can certainly also reduce the risk of intramammary infections. One of the tools to do that is a prolactin inhibitor. At this point in time, that's a bit of a theoretical product because it's not on the market anymore. But the product that is inhibiting prolactin actually leads to a significantly faster increase of lactoferrin, and this is lactoferrin, significantly faster increase in lactoferrin, and thereby reducing the risk of intramammary infections during the dry period. Another one, and I think this is a very interesting one, is the bacterial ecosystems in the mammary gland. Again, using sequencing techniques that I introduced previously, we have learned a lot about the bacteria that are present in the mammary gland. And the typical pattern that we see is a pattern like this. And the message that I want to give here is the diversity of bacterial species. And so every single color is a different species. All the bacterial species are here. They have all sorts of names that are typically not associated with clinical mastitis, very different species. And it looks like the healthier the cow, the more variability we have in these bacterial species. And these are 26 quarters that have a long-term low somatic cell count, multiple measurements of low cell count, and they are culture negative on aerobic culture. And so on the plate, we don't see anything. So these are very clean quarters, and as you can see, an enormous amount of diversity. In contrast, when you have a bacterial infection that we identify as a clinical mastitis or a subclinical mastitis, and we can culture the bacteria on the plate, as you can see here, these are strep uberus cases. There is a predominance of one particular species of bacteria, and that's in this case the strep uberus. And so what you can also say is that an intramammary infection, as we classically found them using culture, is more a dysbacteriosis than it is a specific presence of a bacteria. It means that the enormous variability that we have before has disappeared. We have a dysbacteriosis now with one specific strain, one specific species of bacteria. Well, a few more examples tell us the same thing. This is a challenge study that we did with 4 quarters in a cow. 2 quarters were challenged with E. coli, the hind quarters, and 2 quarters, the right quarters, were treated with antibiotics 5 days later. And what you see if you measure again the microbiome in these quarters, then this is before the challenge. This is the challenge, actually, the purple arrow. Before the challenge, we have this same picture, enormous amount of variability in the different bacterial species. Then we challenge, and it takes approximately 12 hours. We also know that from when the clinical signs are showing. And then we get this dysbacteriosis again. We get an enormous dominance of the E. coli bacteria. Then a few days later, we started treating. We had 5 treatments of antibiotics, and this quarter was actually not treated. And you can see that treatment has an impact. If you glance it over, there appears to be more E. coli here than there is E. coli here. But it's only a stepwise process. It reduces the amount of E. coli bacteria, but it goes step by step. What we also see is that in this particular case, the antibiotics in the non-challenged quarters had relatively little impact on the diversity of the bacterial species. And also later on, when the E. coli is disappearing, you start to see step by step an regrowth, or it's going back to this diversity of the bacteria. And that particular thing is even more highlighted here. This is low cell count quarters, again with the same diversity, both here, and these are 2 high cell count quarters, and this is 2 Staph aureus infected quarters that we measure over time. And if there is no cure, the green dominance remains, the Staphylococcus aureus remains. This quarter does not cure. This quarter is actually cured after a while, and we see that there is a reoccurrence of the diversity of different bacteria. Same here with 3 clinical mastitis cases. This is an E. coli, Escherichia. Lots of E. coli here, very little else. 1 month later, we see a healthy quarter again with a lot of diversity. This is Streptococcus. And again, 3 months later, in this case, we see the diversity coming back. This is a Pasteurella mastitis. And then 1 month later, we see there is no cure, and the Pasteurella still remains the dominant bacterial species with relatively little diversity of bacteria. So I think what we learned from this microbiome is that the enormous diversity of bacteria is the healthy state. And so bacterial or mammary glands are not sterile, they tend to be infected. And the more diversity we have in that infection, the healthier the situation is. And so they are probably able to deal with some challenge. At some point in time, if they go over a threshold, the diversity disappears. The E. coli, they outcompete, so to say, the existing diversity in species, and then the E. coli remains the dominant species. How can we improve the immune response? A few examples at the end. And of course, we can work on the teat end quality. We can work on lactoferrin. At least we used to be able to do that. But another factor that is quite crucial is the negative energy balance. And that is something, of course, in our management, we can deal with the negative energy balance. Immune modulation through the minerals is important, and also the vaccination that I'll show you some examples on. Let's focus a little bit on the negative energy balance. And we know that all cows, in some sense, have a negative energy balance. This is the time of calving. Already before calving, the dry matter intake is so low that cows are using more energy than that they can eat. So we have a negative energy balance. And it takes approximately 10 days, 60 to 70, 10 weeks, 60 to 70 days before the negative energy balance disappears and goes actually to positive. And in this period, the cow is susceptible to ketosis or ketosis-like systems. And we know that ketosis, particularly the presence of these ketotic bodies, so to say, the beta-hydroxybutyrate and others, is a risk factor for mastitis because it is reducing the immune response of the cow. Well, this is a dairy farm in New York that I used to work with. And just in the database that they have, the management system, I made a 2x2 table of ketosis and mastitis. So these are the cows that have at least one case of ketosis, these cows here. And 242 of those also had mastitis, 242 out of 499, so almost half. And of the 1,800 cows without ketosis, almost 1/3 had mastitis. And so there is approximately a double 49 versus 39. It's a 1.5 times as high risk of mastitis in ketotic cows. Well, that is something we see in a lot of farms, and just doing a relatively simple 2x2 table as shown here also helps in making that point clear to the producer. We can also learn that from studies. This is a fairly recent study on the impact of beta-hydroxybutyrate. So these were healthy cows that were infused with beta-hydroxybutyrate. So they were on a drip where they got for several days actually a drip of beta-hydroxybutyrate, and then they were challenged. And so they were challenged with the strep uberis, and the cows on the beta-hydroxybutyrate drip, the blue ones, showed a much more dramatic response to the challenge with Streptococcus uberis, and they showed a significant higher somatic cell count. Actually, the non-BHB cows were able to deal with the infection relatively straightforward. The cell count increased, but not a lot, whereas the cows under the BHB blanket, so to say, showed a very dramatic response and very high somatic cell count. So BHB by itself, even in a otherwise healthy animal, leads to a very much worse immune response of the cow to an infection. Another tool that we used to have is PEG bovigrestin, and BG colony stimulating factor, and it also improves the immune response. I think I'll skip that for the reason of time and just tell you a little bit about the vaccinations, and that's part of the adaptive system. By vaccinating cows, it leads to higher antibodies, and in most cases, what it means in terms of E. coli bacteria, that we have much less severe systems. So again, this is a challenge study that we did. In the control cows, what we looked at is milk production. We got a severe drop in milk production, whereas in the vaccinated cows, they were much better to deal with the incoming infection. They had a better immune response and had much lower loss of milk compared to the non-vaccinated animals. So it reduces the severity. With Staph aureus, as we indicated before in the molecular systems, the transmission is the key. This is a study that we did on the impact of vaccination on transmission. And what we see is that in vaccinated animals, we actually see a 40% reduction in transmission. And so if a cow is infected, it is 40% less likely to transmit to the next cow. So probably lower CFUs and lower risk of transmission. Well, with that, I think I'm coming to the end of my presentation. The key of dealing with udder health on a farm is these three components. First, what is the risk of intramammary infection? What is the impact of duration? Can we shorten the duration? And third is the transmission. And with the new tools that we have, diagnostic-wise, treatment-wise, immune response-wise, I think we have much more ability to keep the farm healthy and have a healthy herd. Thanks for your attention, and I'm giving it back to our chairman, Stefan.
Thank you, Inte. Thank you very much for that very interesting talk. I think we have time now. We have 10 minutes left. We have time to address questions. I see that we already have some questions in the Q&A section. So we have a first question from Eric. Have you new findings about the role of biofilm-coated bacteria in mastitis diagnosis and treatment?
The biofilms. I have not done research on that, but I know there is research done on the ability to produce biofilms. As far as I know, most of that is in vitro work. So we look in vitro whether they are able to make biofilms or not. And I have not seen a lot of that in vivo work. So there is some work done, and some strains are better able to make biofilms than others. But I think it's fairly early in the game to make a real big conclusion on the impact of this theoretical in vitro ability to produce biofilms.
Okay, and what do you think about the impact on possible diagnostic or the treatment of the impact of biofilms?
Well, if what we see in vitro, I have to say it correctly, in what we see in vitro, of course they make a layer which would make them more resistant to cleaning in general, disinfection in general, and probably also or possibly also to treatment. So these strains would then more likely be more persistent. So in my three components, the duration is likely going to be larger if this is indeed happening in the mammary gland as well.
Yeah. Okay, great. Thank you. The second question came from Enrico. From your studies, what is the percentage of contagious or environmental strains for aureus, coli, and uberis?
Yeah, that's a very good question. It depends an enormous amount on what is happening on the farm, and that is actually what I like about this diagnostic system. Is there's very often what you see on a farm also is reflected in what the diagnostic systems are showing. So on some farms you see a lot of E. coli, and then you go to the farm and you see a lot of dirty cows, and you say, okay, it's not strange that we have a lot of different strains, so it's more environmental. And the same with the quality of the milking system and Staph aureus transmission. But it's not always the case. In some cases, you're quite surprised to see what is happening. Particularly with E. coli, I find much more contagious E. coli, long durations and transmission of E. coli strains on farms than I would have expected based on my classical understanding. Actually, with Strep uberus, if you go to Strep uberus problem farms, you probably find about 1/3 contagious and 2/3 environmental behavior. So 2/3 there's lots of different strains, and 1/3 there is a single strain that leads to outbreaks of strep uterus. Klebsiella, it's probably 50-50. Klebsiella is a very specific problem. Sometimes it's the bedding material and it's really bad, but in a lot of cases, actually, you get long-term infections with Klebsiella, and transmission is quite possible. Staph aureus, the dominance is still contagious behavior, so few strains, but there is also a number of farms that have environmental Staph aureus, where you only find one strain per cow, and every cow has a different strain. So what it helps is your own understanding as a veterinarian, but the story that you tell to a farmer and try to educate him on why this is so important on the farm and why he has to do something about contagious strains or environmental strains is enormously valuable to have the pictures connected with it. So you know more, you have a better story, you have a better understanding. Well, the other way around, you have a better understanding and therefore a better story to convince the farmer to make changes.
Okay, good. There's a second question from Enrico, which is related to this one, so I will jump on it. Does a strain identified as contagious in one farm also behave as contagious in other farms, or can it also behave as environmental in other situations?
Yeah, well, I'm pretty convinced there is a strain component to transmission, so that strains are more likely to be contagious compared to environmental. But part of it, as I also indicated in my scheme, if you have a strain that is likely to be contagious, but you are very aggressive in identifying, in treating or segregating that strain, it doesn't have the ability to transmit anymore. And so in that case, it behaves like environment because it only infects 1 or 2 cows. Whereas in another farm where you're not as strict in following up on infections, this strain has the ability to transmit like crazy. So yes, there is a strain-specific component to it. And the Agalactia is a good example. Some of these human strains are not transmitting in cows, and other strains are very cow-specific and are transmittable. But there is also a management component to it.
Okay, good. So another key question from Wojciech in Poland. What do you think has the greatest impact on the resurgence of Strep agalactiae? Could this be related to the reduction of blanket dry-off therapy, as we see in some countries, and that have introduced selective dry coat therapy?
Yeah, that's a good question. What is the reason for it? I used to work on a lot of farms that had, enormous outbreaks of Agalactia, and then they were very easy to find. And you have cows with clinical mastitis, with very high cell counts, and you identify them, and you have lots of Agalactia. If you treat the cows and you improve milking, etc., the Agalactia disappears. And so we thought the problem was over. But if you look much more precise, like culture all the clinical cases or do consistent bulk milk testing, you actually find again, or now in smaller quantities, still the Agalactia present. And as we saw from the Danish data, and I think that's similar to what we find in the Netherlands, is that approximately half of those are more the human type strains, the GBS, probably transmitted from people to cows, and the other half is the more the cow-like strains. So I don't have very hard data or evidence to say that it's because of the lack of dry cow therapy. If it was really the lack of dry cow therapy, I think I would have expected more of these bovine outbreaks. And I don't see a lot of those. It's still there, so there may be some effect, but I think it's a little bit of overseeing it in the last 10 years, and now because of better diagnostics that we start to see them better again.
Okay. There's a question regarding diagnostic test because we talked about a very advanced test, and could you comment on the development and value of co-side diagnostic tests in the understanding of epidemiology?
Yes. So there is some cow-side tests and the color changes, etc., that tell us whether it's gram-negative or gram-positive, and they can be useful in terms of choosing therapy. There is cow-side in a sense of on-the-farm culture. I've seen that a lot on dairies, but it really requires a very good person to do it. There is probably more problems with it than successes with it, but some farms are really good at it, and then they get fairly precise data and the strains or the bacteria that they deal with, and they can adopt their treatment procedures. And if they see an outbreak, and suddenly there is lots of Klebsiella, they can go into much more detail. So on some farms there is good use of cow-side testing, but in a lot of farms it's not very precise. And as a veterinarian, if you really have an interest in that, you can improve that situation enormously by continuously checking for quality.
Okay, I've got a question coming from Germany. I'm not sure you can answer this. Can you give addresses of milk laboratories in Germany which offer this gene sequence test in milk samples? If you have any idea or not. If you don't, we can.
Yeah, I don't. No, I don't know specific.
No evidence of that. So okay.
Sequencing is probably too expensive for dairy farms.
Yeah.
Unless you have a very specific thing. But the MALDI-TOF and the biotyper are very accessible. They are in the sense of 10 to 20 euros a sample, and they give an enormous amount of information.
Sure.
Yeah, and most larger laboratories now have MALDI-TOF systems. Yeah.
So another question, and maybe it will be the last one because we are running out of time. Why is it not yet possible to do a direct pathoproof genome sequencing on mastitis milk?
No, that's exactly what we do. If we look at the microbiome, we sequence the milk and we segregate out the host DNA. We keep it to the 16S, the bacterial DNA, and then we look at all the bacteria that is present in milk. So that is very well possible. The microbiome work that I showed you, and the different diversity of species, that's exactly what we do. Is the sequencing of the bacteria in milk.
Yeah. Yes, maybe it was talking. He was talking about pathoproof as a name of a test, which is only looking at some bacteria, not every kind of species. But as you said, the last advanced tests are able to identify any kind of bacteria.
I think PetApprove works actually on milk. The people that do PetApprove, they are pig people, so they probably do a little bit more respiratory work in pigs or enteric issues in pigs. But the technique, I'm convinced, is also working in milk. Yeah.
Okay, so I've got an answer for Germany. The University of Gießen is doing MALDI-TOF for milk samples. So we have an answer for the previous questions. And maybe if you have 1 minute more to something about Serratia mastitis. What do you think about Serratia?
Say it again. What is the
Serratia mastitis? Serratia marescens, or
I don't get it. What is that?
You can, if you can have a look in the, can you access to the Q&R?
The chat?
Yeah, the Q&R.
I think I'm in a different group. I'm in the host group, I think so.
So, anyway, we're running out of time. Sorry for the three last questions we cannot address at the moment. So thank you again very much, Inte, for the nice talk and the discussion after with the questions. Thank you all attendees for having participated. We were almost 300 at some point tonight.
Nice.
And please stay tuned for the next session. We were able to provide you, as always. We will continue our series of webinars. So good evening or good day to everyone. Thank you again and goodbye.